stem cell and cancer. First of all, Darwin's main dilemma. So while he was proposing this natural selection theory, he had a problem with his honeybees, social insect bees. They have altruistic behavior. As you know, some of these bees, they sacrifice their reproduction in reproductive benefit for the group fitness. So he could not explain how he can incorporate their behavior evolution within Darwin's natural selection. So he proposed that the selection can act on the group, not just on the individual. He wrote that in one paragraph in The Descent of Man. It could not make a mathematical model. In 1964, Hamilton came with a kin selection theory plus a mathematical model. The mathematical model has been here, the skin selection. As RB is more than C, where R is a genetic receptionist key of the recipient of the altruistic actor. As you know, in cancer or stem cell, I will be one. Because in a stem cell colony, I will be always one, where everybody is related to others. That's key. B is the reproductive benefit to the recipient of altruism. The one who received the altruistic benefit will have a reproductive benefit. C, the reproductive cost to the altruist. C cannot be more than the RB. That was the theory. These are the seminal paper Hamilton published in this American Naturalist back in 1963. Since then, there had been kind of an attempt to show altruism in unicellular life. Whether in life when they evolved back during the Precambrian evolution, about 2.3 billion years ago, whether altruism even existed. That was, the question has been drawing many of these evolutionary biologists for decades. So there has been documentation of altruism in bacteria. As you know, quorum sensing or the bacterial biofilm has been considered to be a form of altruism. There's also paper published in Nature back in 2010 by York Age group. They showed that by E. coli resistance against norfloxacin, how antibiotic actually resistant from a bacteria use that biofilm to have a layer for protection. However, there are three things that goes against this. As altruism, to really show altruism, we need to have a molecular mechanism, molecular pathways, how it evolved back during the Precambrian era. We still don't know what can be molecular mechanism selected for group selection. Number two is, number one, cheater genes prevail, always. Even if you have some altruistic genes, the cheater can prevail in a down line of evolution. That has been shown mathematically even by computer simulations. Number two is this, kin selection indicates a group identity. But it's very hard to consider group identity at bacterial or unicellular level. They can have colonies, but do they have a group identity? Do they have a kind of a identity to defend themselves as a colony? We still don't know. Number three is this cooperation is against the survival of the fittest dogma. So that's why the altruism, biological altruism, is still not widely accepted in the field. And in this background, my idea on stem cell altruism evolved from my clinical and philosophical debate. Back in our Hattisatra, where we visited yesterday, with this Vedantic Buddhist and Hindu group of philosophers, the belief in social altruism. I also believe in that. And then during my clinical practice, when I started back in 1994, 1998, I was thinking that how the stem cells, they regenerate themselves after chemotherapy. As you know, when you give chemotherapy to cancer patients, we can kill the cancer cell, but our body's own hematopoietic stem cell, they recover. Remarkable. I thought that kind of recovery after against chemotherapy might involve group selection, group fitness, altruism. So here I gave a talk in this Netherlands Leiden University back in the year 2000. about this altruism theory. And this is an example, like how a single stem cell can recover the entire hematopoietic colonies after chemotherapy in mouse. So I wrote it back in the year 2000. The book is here somewhere. Let's find the book. The squalene book I published in the year 2000. This is squalene is a product of mevalonate metabolism. It's very important, evolutionarily highly conserved. This is the book published in the year 2000. Here I proposed that ancestral stem cells, these are ancestral stem cells, they, because of evolutionary pressure, they evolved to this macrophage system, T cells eventually. But during this evolution, ancestral stem cells I proposed were altruistic. Because if you consider immune system, immune cells, do they have altruistic behavior? Because they sacrifice themselves for the benefit of the entire group. So I thought that if you have to study altruism, we should go for some ancestral stem cell model. And this was kind of, I thought that I used a kind of altruism or group fitness, that if we expose hematopoietic stem cell to cisplatin, is a form of the chemotherapy we use for cancer patient. If we expose this hematopoietic stem cell to cisplatin, we might see some altruistic stem cell trying to protect the other cells. So we developed an experimental model. We published this work in the year 2003 when I was doing my fellowship in medical oncology back in Hospital for Sick Children, Toronto. So we showed that if we expose these hematopoietic stem cells to cisplatin, 2 micromolar, the cells become very toxic. But if we add squalene, there is a recovery of these colony stem cells, and some of the stem cells become very kind of fat-laden, lipid-laden, foam cell type. We considered putative altruistic cell, but we could not really isolate them. We didn't have a biomarker, cell surface marker. So we basically failed at the time. Then I went back to my book here. In this book, I had a figure that I showed that the squalene was present in 3.8 to 2.5 billion years ago in cyanobacterian archaea, their cell member. It was very rich in squalene. And I thought that at the same time, during that time, the world was of hypoxia reoxygenation. The usual life was going through a great period of hypoxia reoxygenation. I thought that if I simulate this microenvironment in the lab, I may be able to activate an old atavistic program of altruistic stem cells, or study the molecular part of cellular altruism. But this is my model I have been using since then. We published this model first in Cancer Research in 2005. Simply, we expose the cells, stem cells or cancer stem cells, to extreme hypoxia, less than 0.1% of oxygen. After 24 hours, we have sudden reoxygenation. The burst of hypoxia reoxygenation leads to burst of reactive oxygen species production that causes this toxicity. And I thought that maybe simulating the microenvironment of primitive earth and then activation of ancestral stem cell altruism. Using this model, interestingly, Dr. Kenji may be interested now, we found this a pathway where MAP kinase is involved. The cancer cells, when exposed to this extreme hypoxia stress, they activate the VEGF-FLT1 signaling that activate MAP kinase ARC1-2, then the BSL2 and also BFGF. HIF is also involved. This is where Masabe Mishibo was he was a collaborator. And his student Vikash Yoshida came to work with our lab at that time. So we found we were interested because MAP kinase is a very ancient signaling pathway. Kind of phylogenetically conserved, as Dr. Kenji might know, from yeast and even earlier kind of kingdom of life. So also VEGF-FLT1 is also very ancient. HIF1 is very, very primitive, at least 900 million years old pathway. So we thought that maybe this VEGF, HIF, MAP kinase R plus HIF program could be a molecular mechanism of stem cell altruism. That was coming to our mind back in 2005. And at the same time, what we found that when we expose some cancer cells to cisplatin. Some of these cancer cells become cancer stem cells by activating VEGF-FLT1 signaling pathway. They activate this ARC1, phospho-ARC1 2. You can see in the cells of osteosarcoma cell that has in this SP means the site population cells. As you know, site population is a marker for the cancer stem cells. They activate the FLT1, also the ARC1-2. So even in a non-SP, they can activate this pathway. So MEP kinase FLT1 is involved in enhancing the stemness of these cancer cells when you expose them to cisplatin, suggesting that this could be a form of cancer stem cell altruism. We published this work back in 2008 in Oncogene. We could not really demonstrate the altruistic behavior at that time. So I come back to this hypoxia. My main question was this: when you expose the stem cells to hypoxia, what will happen to p53 MGM2 oscillation? In stem cells, p53 is highly sensitive, it's a wild type. The little bit of reactive oxygen species damage, p53 is activated, cells undergo differentiation errors. So what we did, we exposed human embryonic stem cells to this system of hypoxia reoxygenation. We thought, let's consider human embryonic stem cell to demonstrate whether we can find kind of evidence of stem cell altruism. This is our key paper on that. As you know, this is a human embryonic stem cell colony. It's a BG01 cell line, of course, and after 24 hours and 40 days of reoxygenation, almost all cells differentiate die. As you can see, DCFSG is a marker of reactive oxygen species. It goes up on day 4 of reoxygenation, p53 goes up, E4 alpha goes down, because this is a very acute gene. After day 4 it goes down, E2 alpha stays similar. And when you look at this cell after flow cytometry, some of this population here, there is a population of SSEA3, they still remain undifferentiated, and there's a population of ABCG2, it's a marker of drug efflux protein, ABCG2. So we sorted this population to make its story short. And we found that this ABCG2 positive cells are p53 deficient compared to ABCG2 negative. They express HIF2 alpha very highly. And when you look for this protein using ELISA, we found that p53 is continuously downregulated at least for 2 weeks. HIF2 alpha is upregulated. And eventually we found that in this, the p53 and MDM2 oscillations are altered. This is a huge paper, I just showed just that only. Suffice to say that in these cells, in ABCG2 positive cells, the cells manipulated that p53 MDM2 oscillation. So for 2 weeks, these cells can remain p53 deficient. So they can tolerate genomic stress. And they secreted squalene and glutathione. When you take this conditioned medium from these cells, they protected, they exerted cytoprotection to the other embryonic stem cells. After 2 weeks, these cells died because the p53 MGM2 oscillation come back to normal states. p53 was activated, cells undergo apoptosis. So we proposed then that that this HIF2 alpha suppression of p53 only for 2 weeks could be a molecular mechanism of stem cell altruism. It was accepted at the time. The reviewer allowed us to use the word stem cell altruism for the first time. And because this is the kind of a summary of the results, that these cells which actually survived this hypoxic stress because of altruistic stem cell, they re-established their colony, they re-established their self-identity. Because the clonal heterogeneity of this embryonic stem cell, we demonstrated remain the same. The self-identity maintained. So Stanford University, because they huge publication, huge report, they come with a press release that finally that in eukaryote cells we can demonstrate a molecular mechanism of altruism. But our findings shows a different from of Hamilton's theory. We did not in the Hamilton theory explained by RB by less more than C is here. We found a DP factor, a defending power. The defending power is this: that p53 deficient cells who have that, they gain the power. Because once you reduce p53, the cells develop fitness gain. But they had a defending power, and they sacrificed that fitness. So it is compared to honeybees different, because those the bee worker, they have no power. They're very simple kind of worker. Sacrificing their fitness versus sacrificing fitness of a P53 deficient embryonic stem cell is different. So we thought that in our case of stem cell altruism, you have to be add a factor dp. So this is we revised the formula. RB plus B should be more than C plus dp. Then eventually we published models of this altruistic stem cells. First in 2003, as I explained, then cancer stem 2008 we published, 2012. In 2013 we showed that mycobacterium tuberculosis can manipulate this bone marrow altruistic stem cell to remain dormant. We published this in Science Translational Medicine, and then come back to our cancer. But this is my interest as a medical oncologist. In cancer, as you know, is considered as a classical example of Darwin's evolution. Clonal selection. Every cancer cell fights for their fitness. The mutation drives the selection process. This is our dogma. But we did not really consider that it could have a collective difference as well. So how do you explain group fitness in terms of cancer? Many times we know that cancer has some phenomena like dormancy could not be explained by this clonal selection. If the cells are driven by mutation, or how they can remain dormant for years, remain quiescent. That's why we have the cancer stem cell hypothesis. That cancer may be hijacking stem cells in a stemness program. And I argued that, well, if this is the case, cancer may be also hijacking stem cell altruism mechanism. So we wanted to find out whether the hip to stemness pathway is activated in cancer stem cell, and if they're altruistic as well. So we first published this paper in 2019 showing that the same pathway we found in embryonic stem cell, hip to stemness pathway is activated in a group of cancer stem cells in a MYC-induced lymphoma model. This model was Gene Felser, he was mentor Stanford. This model was actually a model, mouse model was first established by Michael Bishop, who received Nobel Prize for discovering MYC. I used his model, very robust model. We showed that there is a population of cancer stem cells that remain dormant and they exhibit altruistic behavior by activating the same molecular pathway to VEGF as activated by human embryonic stem cells. And then this is our mechanism. We did a lot of molecular assay, but this is if to make, they have this, the cooperation, they activate GSH, glutathione, they suppress P53. So overall, P53 is temporarily transiently suppressed in certain cancer stem cells. They become high fitness and they sacrifice their fitness for the group fitness. Then this is our primary mechanism. The make it to stemness switch pathway. Stemness could be altruistic stemness, that some of the stem cells can transiently suppress P53 by manipulating this VEGF, MAP kinase, to make pathway, this up the downstream pathways. Then during that time, they could be altruistic. And so it seems that that could be also a mechanism of malignant transformation. We have data, we have certain manuscript under review. We have shown that in oral cancer, at least during the carcinogenesis process, the stem cells that become altruistic, they can actually choose to become cancer stem cells because they have deficient P53. If the P53 ambient alteration, the oscillation remain permanently suppressed, then the cells remain P53 deficient. Then they can kind of become a part of the selection for a mutation gain. So it could be a mechanism that altruism could be a doorway, could be a big bang for cancer genesis. And for this pathway of stemness transforming to altruistic stemness. Then back in 2019, we also showed that oral mice microbiome, microbiome on tumor prostate is becoming very big topic nowadays. The bacteria manipulate the cancer cell. They can reprogram cancer cell to activate the stem cell tooth. That was published in 2019. Then in July 2021, we developed a model of the coronavirus infection with TB infection of mesenchymal stem cell in the lung. This was done by Letika. She's sitting here, and this is the overall scheme. Here, I want to spend 1 minute here. So these are mesenchymal stem cells recovered from the lung. We developed a mouse model of dormant tuberculosis. Already we established that in 2013, 2015, and 19, we have series of paper. Suffice to say that in that model, we can keep the bacteria, Mycobacterium tuberculosis dormant. Once you add streptomycin, the dormant become fulfilled bacteria. So using that, when you actually co-infect that mice with the coronavirus strain MHB1, MHB1 is a well-known model for the liver carcinogenesis. It's been a long time. It's also a coronavirus model. So when you co-infect them, the infection of MHB1 virus, then these cells reprogram. They activate the altruistic stem cell pathway by enhancing HIF2α, suppressing p53. And the secret factor we still don't know, but they include also the squalene also glutathione higher. And in alveolar stem cell, they exhibit altruistic behavior. The cells rapidly proliferate, these mesenchymal stem cells. When this stem cell proliferate, also these dormant bacteria that are hiding inside this stem cell, they also replicate. The dormant bacteria then release from the cells, they infect the lung. So this is how we show that how bacteria can manipulate the stem cell leaves to remain dormant. And then when there's a co-infect with a virus, they can cause reactivation. Based on this data, we predicted that after coronavirus, the TB infection might rise globally. So our paper was considered for a press release by Elsevier or by the AAAS, American Advancement Society. The press release is somewhere here. So yes, our prediction came true because TB infection has risen manifold now in the world after the coronavirus. Maybe this one of the mechanism we still don't know, but this is a very good model where we showed stem cell altruism in the mesenchymal stem cell in mice. And then we showed back in 2012, it was published in Frontier Immunology, that when you take cancer stem cell infect with BCG, is a form of attenuated TB vaccine. BCG has been used in Japan before, also Indonesia is now used. BCG is a vaccine, is also anti-cancer therapy for bladder cancer. When you infected this cancer stem cell with BCG, these BCG manipulated this cancer stem cell to become altruistic. But this cancer stem cell underwent altruism, they died. What they did, when the bacteria was replicating inside this cancer stem cell sent a signal to other cancer cells. They released a factor called H-MGB1 p53 complex, that signal. That bacteria is within me, replicating, so I'm going to die, others should die too. This signal is called bystander apoptosis. This is an innate immune mechanism of macrophages. So macrophages, when they infected with bacteria, any pathogen, in an area of tissue, then one macrophage will signal to the other macrophage that you should all die. It's called bystander apoptosis. They release that signal called H-MGB1. We found that in cancer stem cell, BCG can activate that signal. And this death signal actually activate the altruistic behavior because they lead to this altruistic cell death. This was published in 2022. And recently, last year exactly, we published another paper in Frontier Immunology that we have isolated this so-called altruistic cancer stem cell phenotype from a patient, they're suffering from oral cancer, head and neck cancer. I will show some data that will end my talk. Lekiga was, this is her also PhD paper. We have, our group is here. Here is this data one. This is ABCG2, this is FCAM. FCAM is an a marker, dual marker for head and neck cancer stem cells. We have taken this from the patient blood, from the circulation. We have collected the peripheral blood mononuclear cells, subject to the flow cytometry analysis. Patient 2 and 6, you can see this subpopulation of FCAM positive ABCG positive cells. And we also fed this patient with the P-monodazole. Those who are working on hypoxia will know that P-monodazole is a marker of hypoxia. So if you fed the patient with P-monodazole tablets, then any cells in the body will be marked with this P, the hypoxia. So we found these cells, when you do the flow cytometry, the immunofluorescence, they have FCAM positive, ABCG positive cells are hypoxia because they are P-monodazole positive. Suggesting that as if these cells are released from the primary tumor, which are hypoxic, containing this FCAM positive, ABCG positive cells, we found they're altruistic because they first all this gene expression, a little bit data, if to make laser receptor on here, 6 are all activated, those are the phenotype marker for altruistic cancer stem cell we already identified. And these cells are transient because they only proliferate for about 2 weeks, then they all undergo altruistic cell death. So what we did, then we took this, the patient primary tumor. In the primary tumor, so in the primary tumor, you have to come this way to show the data though. FCAM positive, ABCG positive cells. There is a small fraction of the cells, we can sort them out. When you do the flow cytometry analysis for the P-monodazole, those are P-monodazole positive, suggesting that in the primary tumor, we can isolate hypoxic cancer stem cell expressing FCAM positive and ABCG positive cells, and these also cells have this altruistic phenotype, altruistic stemless phenotype expressed. We have selected about 15 genes. We have kind of characterized them to be a altruistic stemless phenotype back in our 2019 paper. So these are the genes highly expressed. Basic receptor 1 is highly expressed. We have not yet mapped these cells, whether they activate MAP kinase pathway or not. This is our ongoing work right now. So this is our result, and we do have a manuscript under review, exciting. We have shown, and he is not here, he has gone to Dibrugarh city for a presentation. Partho, he is not defending his PhD this year. He has shown that Fusobacterium nucleatum is actually bacteria present in oral cavity, also in our gut. Fusobacterium bacterium could be manipulated, reorganized, or reprogrammed by this altruistic cancer stem cell to become highly virulent. It shows that when our cancer patient exhibit stem cell altruism, this patient died very soon. We have done a survival mechanism. As soon as we see these patients in the circulation, this kind of cell, they only remains only for about 3 to 4 months. They die of septicemia, severe bacterial infection. That led us to hypothesize that there may be a crosstalk between bacteria and then stem cell altruism, because as you know, stem cell altruism could have been a very primitive mechanism of survival back in the evolution when bacteria and unicellular life coexisted. So we think that cancer is probably reactivating an atavistic signaling mechanism of survival that existed during origin of life, when the earth was very toxic, full of radiation, hypoxia. It seems for cancer, even though we were targeting them with radiation, also chemotherapy toxic agent, they can activate those primitive altruistic stem cell signaling for survival, defense. So this is the ongoing work of our lab. And this is our tower lab back in Lowell, University of Massachusetts. So team. So this lab we have seen, that's their lab there. This lab was originally in Forsyth Institute of Cambridge, part of Harvard Medical College. Back in 2018, I decided to move out of that, make an independent non-profit organization called the KaviKrishna USA Foundation, so that we have these two same labs, America and India, and so we do not have to go for this intellectual property theft, because Harvard told me that you cannot run Indian lab. They're stealing our American idea if you do so. I said, okay, and move out. So now we have an independent lab, it's a good team. We're developing a biosensor to sense altruistic stem cells. So Lekika is, now going to the lab. She's also working on with the University of Massachusetts. She found a postdoctoral position there. She's supposed to go on.
Once the war is with
Our flight was in here in 19th March. It's got cancelled. Anyway, so this is what about the lab, and thank you for listening. And my comment is that Professor Kenji is here. It's a very good opportunity today. We can talk about this stem cell altruism, where one of my Japanese, mentor, Masashi Shibuya, who encouraged me back in 2005, and also his fellow Rika came to work with us. We came to know about Toshio Suda from Kyoto University. Toshio Suda's friend, Dr. Ohnida, lab students. Markus is here today, right? Great to know. And Professor Kenji is here. His pioneering work on yeast could be a good model for studying altruism, yeast. With that kind of a collaborative idea, I end my talk. Thank you.
Thank you very much.